concentration of the superlimited. And as a reference, we have fibrils that are kind of this white color. That means that obviously there's no dye bound. The advantage of this titration protocol is that we don't have any DMSO in the sample. That's the first thing. Second thing is that we can reuse our sample multiple times. So after sedimentation, we can resuspend the pellet. We can add and titrate the BSA adapter solution on top. We wash, we can pack it, then we can again unpack it and resuspend, and we can redo this titration several times. And this allows us so that we always look at the same fibril polymorph. We're not seeing any spectral changes from one step to the other. And we can also do a concentration dependence, right? So we can slowly increase the concentration of the dye in order, the idea is to get a binding curve in the end. And you see that from one step to the other, we don't lose a lot of fibril material. So the intensity is more or less reproduced. Maybe we have like a loss of 5 to 10% per titration step. Then we recorded the first carbon-carbon correlation experiments, and we were rather disappointed because there is basically no large chemical shift changes in the presence of the adapter molecule. And then this could have two reasons. So we assigned it sequentially, and obviously, as you know, for the A-beta, the N-terminus is largely disordered. The C-terminus is structured, and it could potentially be that the small molecules bind to the N-terminal half of the peptide that is not structured and not assignable by solid-state NMR. Or alternatively, it could also be that carbons are just too insensitive to sense small chemical shift differences between the bound and the free state, because carbon have a low gamma. They're a little bit further away from the binding site because they're decorated with protons. Protons should be then more sensitive to detect changes in the chemical environment. And on top, we evolve carbon-carbon J couplings. This makes the lines relatively broad. And we thought maybe the reason for this is that carbons are just not sensitive probes to detect changes of the chemical environment. I just want to focus on this spectral region again so that you can see that the spectra that we obtained in the presence and absence of the small molecule look almost identical. There's very little change. And yeah, and we couldn't really go on. And at this point, before I show you the proton detection experiments, I wanted to show you some reference experiments that we did a few years earlier with this sulindac sulfide molecule that we investigated together with Gerd Multhaupt from Montreal. This is a molecule that rescues cells from the neurotoxicity that is exerted from the A-beta fibrils. So for example, here, when you look here, under these conditions, 50% of the cells die in the absence of the small molecule. And when you add the small molecule, here the black curve, then you see that only 20% in the cell culture are dying, and so to speak, 30% are rescued from toxicity. And when we titrate sulindac sulfide to A-beta, then we actually observed large chemical shift changes. So for example, this alanine 21 signal totally disappears and a new signal pops up, which would tell us that each and every peptide, or it's to each, a small molecule is binding to each and every peptide in the fibril, because otherwise you would still see some intensity at the original peak, or you would get a smear and broadening of the resonances, but it's not the case. So we see that the peaks they're recorded in the absence disappear, new peaks show up. And at that time, we looked at chemical shift perturbations. Sulindac sulfide contains a fluorine, and we also did a fluorine redo experiment to really localize the direct binding. And we identified the binding site to be close in the vicinity of phenylalanine 19, but also involving glycine 33. So we think it intercalates and somehow binds into this groove that is spanned up by these glycine residues, which are obviously providing a cavity. There's no side chain, so they provide a cavity where potentially a small molecule can bind. So this was just a little excursion for sulindac sulfide. Now let me return to these PET tracer experiments. And these spectra show the proton detected experiments that we did on deuterated amyloid fibers using a 1.3 millimeter rotor and rotating the sample at around 55 to 60 kilohertz. And the spectra that I will show you come in two flavors. So on one hand side, I just show you the normal proton-nitrogen correlation experiments that you're used to from solution state NMR, just proton and nitrogen axis. But on the other hand, it turns out to represent the data also like a proton-carbon correlation. We use dipole interactions to transfer to the next neighbor, C alpha carbon. And we plot here the C alpha chemical shifts, and this is an advantage because you can nicely see the glycine spectral region, and you will see that this is an advantage in the following. And what you also see is, when you have a closer look at those spectra, that we don't get a single set of resonances, but for certain residues, we see two sets of resonances. So for example, this glycine 33 comes in two flavors. Here's a peak, here's a peak. This peak is even broadened, so we think that there are potentially three conformers that are underneath glycine 33. The same for alanine 30. You see here a peak, another one comes here again. And that means that protons are very sensitive to detect small conformational changes in specific regions for the amyloid fiber. Now, next slide shows the experiment that we did in presence of Pittsburgh compound B. On the left is the reference spectrum. Again, here I show you the proton-carbon correlation experiments. You have here the glycine spectral region. Here is in the presence of the compound, and here's the superposition. And you see that there are quite some changes occurring. And let me focus again on these two regions. On one hand side, I would like to focus on the glycine spectral region, and on the other hand here on the alanines. And when we focus in, then you see here in the presence of the small molecule, the heterogeneity is lost. So here we have these three peaks of the first conformer, the second conformer of alanine 30, but in presence of Pittsburgh compound B, that this heterogeneity is lost, and it seems like that the small molecule suppresses conformational heterogeneity and maybe even stabilizes the fiber state. And here for the glycine spectral region, there are large chemical shift changes involving glycine 25. So the original peak is here, and then the peak moves to this position. And the same for the other glycine, moves from this position to this position. And we confirm this obviously by sequential assignments. Most chemical shift changes take place in the loop region involving this GSNK motif, and another large chemical shift takes place around glycine 33. And this is again reminiscent to these experiments that I showed earlier for the sulindac sulfide. When you look into the structures, and here's Rob-Tico structures, but this also applies to other cryo-EM structures that have been solved recently, you find relatively large cavities in those structures that can potentially accommodate a hydrophobic small molecule. So here's the structural model from Markus Fendrich's group, Marius Kolmer, and here's the cryo-EM structure from Gunnar Schröder and Lothar Krämer. And I just indicated here with these gray balls, cavities where potentially a small molecule can bind. And again, when you look there, so we see large chemical shift differences in this GSNK loop. This is here. So we believe that Pittsburgh compound B occupies this cavity in the bound state, but it can also potentially accommodate and bind to the cavity number 2. Now, at last, we are also trying to get resonances for PIP itself. Therefore, we N-methyl labeled the compound and also incorporated here methoxy C13 group. And the dashed lines indicate the region where we would expect the resonance. However, under the conditions where we tried, we could not see any peak. And this could have two reasons. On one hand side, it could be that the resonances of PIP are broadened because of chemical exchange. It might go back and forth between different binding sites. Or alternatively, it could be that the peaks are broadened because of dipole interactions, because we are only rotating the sample at 55 kilohertz, and this might not be fast enough in order to detect the peaks. You can actually rule out that chemical exchange is the issue, because in this titration protocol, we did not only use a 4-fold excess of the molecule, but we went up to a 20-fold excess and still could not detect the small molecule. And in the following, in the remaining part of the talk, let me quickly show you our results that we obtained and our theoretical considerations why we think it's dipolar broadening that prevents the observation of these peaks. So here we switch to a model protein. That's the alpha-spectrin SH3 domain. It's a crystalline protein that is perdurated and only selectively protonated at valine and leucine sites. And we carried out Simpson simulations in which we assume a 9-spin system. So we looked at each and every methyl, screened the environment, took the closest methyl groups in the X-ray structure, and calculated, so to speak, the proton line shape as a function of rotation frequency. And what you can see here is that for certain groups like leucine 8, at a rotation frequency of 100 kilohertz, you reach 80% of the maximum possible intensity. However, if you do the same for valine 9, and then at 100 kilohertz rotation, you only reach an intensity that corresponds to 20 to 30% of the maximum intensity. And we can do this for each and every methyl group. And you see here in red are the experimental data. We compare them to the simulation. You see that simulation and experiment match rather well, maybe with the exception of this one residue, valine 8, which is involved in crystal packing. And therefore the simulation might be a bit off. So in reality, the dipolar coupling that this valine sees is larger than what we assumed in the simulation. And we define a value, nu 50. This is the rotation frequency, the MIS rotation frequency that is necessary to achieve 50% of the maximum possible signal to noise. And what you see is that across the sequence, for some methyl groups, the rotation frequency of 20 kilohertz is sufficient in this deuterated system to achieve 50% of the maximum possible sensitivity. But for other methyl groups, maybe 300 kilohertz is necessary to achieve this. So the conclusions for us is that we still need faster spinning probes, where we have a perdurated system. So we think that actually rotation frequency should be fast enough, but still it's not sufficient. And this brings me to the summary. I hope I could show you that BSA can be employed as a carrier to titrate hydrophobic molecules to amyloid fibrils. We observe no chemical shift changes for carbon-based experiments, but subtle small chemical shift changes in proton-detected experiments. In the future, we would like to repeat these experiments using a 0.7 probe, so we have higher frequencies available, and we hope to be able to detect the PET tracer molecule. And we also are able to prepare different polymorphous forms of A-beta fibrils that have different spectral characteristics. So both samples are enriched and they are pure, so to speak, for this particular polymorph. And we would like to know if the small molecule can bind to morph 1, but not to morph 2. And this way try to find out what are the binding characteristics of the dye. So this brings me to the end, and I would like to thank the people who were involved in the work. This, what I talked about, was basically the PhD project of Zhong Yu. The experiments that have been done on sulindac sulfide were done by Elke Brade. The MAS frequency-dependent experiments were done by Kai Xu, Rediman Sarkar, and Stenik Toschner. And I would like to thank you for your attention.
Okay, thank you, Bernd. Looks like Andy has a question.
Hi, Bernd. Great to see you. Very interesting. I'm curious about the relative affinities for your small molecule of choice with BSA and with the fibrils, because in doing this sort of carrier swap, you have to be at least aware of that balance. Do you have some idea of those numbers and how that goes?
We don't know anything about the affinity of the small molecules to BSA, but from in vivo experiments, so Hans-Jürgen Wester did affinity to amyloid plaques, and these affinities are in the nanomolar. So we are relatively certain that it's going to be direct over.
Great, thanks.
Rieko?
Yeah. I'm from University of Pittsburgh. Since you mentioned about Pittsburgh compound B, I thought better to ask one question. And of course, in University of Pittsburgh, there is a strong Alzheimer Research Institute, and such a research is going on. And but I don't know anything about those amyloid. Since you mentioned this compound is very hydrophobic, not soluble to aqueous solution. When the compound binds to amyloid in the brain, is there any lipid or other factors affect the retention of the compound?
That's a good question. I think experimentally, what people do is when they inject this into the bloodstream, and then this is done at extremely low concentration, because for PET imaging, you need picomolar concentrations. So there's basically, then solubility is not an issue. And I think experimentally, one has also seen that it only binds to the plaques and is not found anywhere else.
That means that, for example, when such an amyloid accumulation in the brain is compared with the distribution of the compound bound site, are they completely agree or depends on the brain environment factor?
I'm not sure if I understand the question correctly, but well, you ask whether it binds specifically to all fibrils or selectively only to a subset?
Specifically to certain environment in the brain where there's amyloid, or there's amyloid but slightly different brain composition, something like that, a soluble part or insoluble.
I think this is exactly what the chemists would like to find out, right? And obviously they would like to have small molecules that bind to precursors, not to fibrils, mature fibrils, but to some oligomeric intermediate states. And and it could be that other factors also affect the binding. It could be that it only binds to a subfraction of all amyloids in the brain. That's what I try to say, that they maybe only binds to a certain polymorphous form. And that would be the goal, to find out maybe certain fibrils or aggregates have a particular neurotoxicity. And these are the ones that you would like to identify, right?
Thank you very much. It's interesting. Yeah.
Actually, I had a really quick question. So if I understood the structural models correctly, I mean, it seems like that in some cases, these small molecules are incorporated like in the interior of the hydrophobic cavities. And I just was curious if you have any kind of comments or speculation about the mechanism of how these molecules get in there, if this is correlated with some breathing motions or dynamics in the fibrils.
That's a good question, right? Well, from an entropic point of view, you would not be able to understand, or enthalpic point or energetic point of view, you would not be able to understand why they can go inside. But obviously, the binding inside would be much more favorable, right? So the energy would be, well, the delta G should be much lower if they bind inside, because then they're totally shielded from the environment. If they bind on the surface, then that they are still exposed to some kind of solvent, and which would not be favorable binding event. And how this goes mechanistically, it's not known. I only know that when you do fluorescence microscopy of fibrils that have a certain color and mix them with fibrils of a different color, let's say they're green and red, and you mix those fibrils, then they become yellow. So it means that there must be a certain degree of dynamics that allows exchange of individual peptides. At the moment, I also cannot imagine how this would work, but this is what people observe, fluorescence microscopy people observe experimentally.
Right, so with that, let's thank Bernd again for an excellent talk. And the next speaker in the session will be Sucharita Sarkar from University of Delaware, and she will talk about, I hope, the structural basis of HIV-1 maturation inhibitor binding and activity by solid-state NMR.
Can you all hear me? Okay. Good morning, everyone. I am Sucharita Sarkar. I'm a graduate student in Tatiana Polinova's lab at University of Delaware. And I'm very excited to say that this is my first in-person conference talk. So I would like to thank the conference organizers for giving me the opportunity to share our research. So today I will discuss the structural basis of HIV maturation inhibitor binding and activity by magic angle spinning NMR. And I'm going to discuss something really unique from NMR standpoint. I will tell you three things. First, how to solve atomic resolution structure of macromolecular crystalline protein assemblies in presence of small molecule. I will also tell you how to detect direct protein ligand interaction of natural abundant small molecule in protein assemblies. And finally, I will discuss the power of phosphorus NMR for screening the small molecule binding and activity inside proteins. And to do that, I will use a system of HIV immature GAG intermediate lattice in presence with maturation inhibitors. So why are we interested in this system? I want to capture your attention here because this is a really unique system where cryo-EM or X-ray or any other method cannot determine the asymmetric drug binding inside a protein at atomic level, but we can do by NMR. So let me tell you about the system in details. So as we all know that HIV is a deadly retrovirus and widespread, and since HIV were reported 40 years ago, 79 million people have been infected with HIV and more than 37 million people have died due to AIDS-related illnesses. In year 2020, 1.5 million people were newly infected with HIV. So despite years of research, HIV is still incurable and it remains a difficult target for vaccines. So it is essential to understand the structural biology of HIV to improve and develop new therapeutics. So in HIV life cycle, it is to become infectious, the immature virions must undergo maturation. And this is a really important step in HIV life cycle. Despite years of research, there are still open questions about HIV maturation. So let's look at the maturation step. So HIV virus has 9 genes, which encode 16 proteins. And today we will be looking at the GAG polyprotein, which is encoded by GAG gene. GAG polyprotein is a principal structural protein, which is consists of matrix, capsid, spacer peptide, nucleocapsid, and other proteins. Upon budding from an infected cell, the shell of the immature virion, it comprised of several hundred copies of GAG polyprotein. And during maturation, it goes through a cascade of cleavage event by the viral protease. And this is a programmed operation of molecular switches, which triggers both local and global structural rearrangement. And this leads to the formation of the mature virion, including the formation of the viral conical capsid core. And we are studying the last step of this maturation step, which is the CASP1 maturation switch. And our protein is the C-terminal domain of the capsid protein, along with this SP1, which is a 14 residue polypeptide. So I'm going to call the protein as CA-CTD-SP1 from now on in my talk. So CA-CTD-SP1, it forms immature type of lattice. With the CA-SP1 switch region, it forms a 6 helix bundle. And the CTD forms a cup, so it's like a goblet-like shape. And CA-CTD-SP1, it assembles in vitro into flat sheets with this subunit organized into hexagonal lattice. Okay, let's look at the maturation inhibitors now. So this is a new class of antiviral drugs which are known to interfere with the HIV maturation by binding to the immature type of lattice at the 6 helix bundle. And this disrupts the final step of maturation, which is the CASP1 maturation switch, and it prevents the formation of the viral conical capsid core formation. So what does it do? It forms non-infectious, immature type of virus particles, which are incapable of infecting other cells. And we are working with bifidimab, which is the first GAG targeted compound, first in the class of maturation inhibitor to reach clinical trial. And previous studies have shown that bivirimat binds at the 6 helix bundle, but then why are we studying it? Because in other methods such as cryo-EM or X-ray, the flexible sp1 tail, it was not detected. And also the structure of this asymmetric inhibitor binding inside the 6-fold symmetric pore, it cannot be determined at atomic level. As we can see here, the binding orientation were not determined at atomic level by other methods. So these are very promising class of inhibitors, but there are some HIV mutations which causes resistance to BVM. And our collaborator at NIH NCI, Eric Fried and his group, they are right now working on designing better maturation inhibitors. And in order to do that, we need to understand the binding mechanism of maturation inhibitor to understand the resistance mechanism. So to do that, we have studied these two specific mutations, which is sp1A1V and sp1V7A. I will tell you about this in details in my slides later. In addition to understand the maturation inhibitor binding inside the CSCTD-SP1 immature type of lattice, we have also studied the interplay in between the protein and this cofactor, which is called IP6. IP6 is naturally occurring in human body, and it is used by the virus to promote and drive the assembly of the 6-fold symmetry and the assembly of the immature type of lattice. And previous studies have shown that IP6 binds in the immature type of lattice near the binding region of the 6 helix bundle where inhibitor BVM binds. So it is interesting to understand both of this small molecule binding and interaction inside the CSCTD-SP1 immature type of lattice, and this is what we are going to do in both wild type and resistant mutation. So for our project, the CSCTD-SP1 immature type of protein assemblies, it was prepared by our collaborators at University of Virginia, Barbie Pornillos, Owen Pornillos, and Kanil Zadrozny. So as I mentioned before, that CSCTD-SP1, it forms immature type of lattice in vitro. And it is morphologically different than the CASP1 tubular assemblies, which forms mature type of lattice. And in 2013 to 2015, students from our lab and our collaborator, they have studied the mature type of lattice, which is the CASP1 tubular assemblies by NMR. And when we started studying the immature type of lattice, we have compared these two assemblies. And as we can see here, both of these assemblies, they yield remarkable spectra of high quality. But if we look at the various regions of the spectra, we can see the chemical shifts are very different in between these two assemblies. And what does it indicate? It indicates the local difference which is associated with the mature type of lattice and immature type of lattice. So it was not possible to assign the chemical shift of immature type of CTD-SP1 lattice just by comparison. So we have done the assignment from scratch. And to do that with my co-worker, Roman Zadrozny, he is in the audience, and with our UDNMR facility scientist, Caitlin Quinn, we have carried out the solid state NMR experiment. And I'm glad to say that we were able to complete 96% of the chemical shift of the protein. But most interestingly, we are seeing this terminus residue here and the flexible sp1 tail, which is first time detected by NMR using various type of NMR experiment, as you can see here in the backbone walk. So in this slide, I'm very excited to say that we were able to obtain numerous unambiguous inter-residue distant restraints from uniformly carbon C13 and N15 labeled sample. And given the peaks are very well resolved, so we were able to obtain more than 50 distant restraints for most of the residue inside the protein. And most importantly, we are seeing these interchain and interhexamer distant restraints, which are important for structure determination of higher order assemblies inside the protein. So far I was able to assign or work on this assignment distant restraints more than 3,000, and which means 30 restraints per residue. And I'm very excited to say that this is the highest number of distant restraints that were assigned for MAS NMR structures, which is reported in the literature for the proteins with more than 100 residues. So here I'm coming to the most important part of my talk. One of the most critical questions is how we determine the binding interaction of natural abundant small molecules inside a protein. So we have used proton correlations from the small molecule to the protein. And to achieve this, we have used fractional deuteriation and a suite of dipolar-based head core experiments. So first of all, for the protein with only IP6, we can see the proton correlation from the IP6 to the residue at the binding site, and we can detect this direct correlation in our NMR spectra. So what happens when we also have BVM? In addition to the IP6 and protein correlation, we were also able to assign numerous BVM and protein correlation, which first of all determines the orientation of BVM inside the six-fold helix bundle. And also it determines the simultaneous binding of both cofactor IP6 and this inhibitor BVM inside the 6-helix bundle. And these are the most essential sets of experiment that gives us the structural information of natural abundant small molecule inside a protein. And in addition, my coworker Roman has performed this phosphorus NMR experiment where we detected that IP6 undergoes dynamics inside the protein, but in presence of BVM, IP6 is rigidly bound. And I will discuss this just in few more slides. Okay, so what do we have so far? We have the protein-protein distant restraints. We have the correlation with the small molecule and the protein, which gives us the BVM protein distant restraints and IP6 protein distant restraints. So with all of that, with my coworker Ryan Russell at UD, we have calculated this system, which is a hexamer of hexamer protein assembly in complex with the small molecule BVM and IP6. So what do we have from the structure? We can visualize the protein in presence of these two small molecules, and remarkably, we can see that the sp1 tail here is well defined in our structure. And finally, we have detected the small molecule binding site at atomic level. So with all of this, as we have this large number of long range distant restraints, which gives us a very tight pairwise RMST of 0.6 angstrom for these structures. So now here, I want to capture your attention because we have observed something very important and exciting in presence and absence of inhibitor BVM. So in presence of the inhibitor, we see a large conformational change in the junction helix, which triggers the tightening of the 6-helix bundle at the pore, and also it rigidifies the IP6. And as I pointed out before, that X-ray or cryo-EM cannot detect the atomic level picture of the asymmetric binding of small molecule inside a symmetric pore. Here is the beauty of NMR. We see multiple correlations in presence of BVM, which indicates the asymmetric binding of a small molecule inside a protein. And also this leads to conformational heterogeneity of the protein side chain near the binding site. Okay, now let's switch gear towards the HIV mutation, as I was telling before. So we have studied these two specific HIV mutation, which shows resistance to the BVM, which is sp1A1V and sp1V7A. So these binding mechanisms are very distinct and subtle. One change in the amino acid sequence, it changes the dynamics and interaction of the drug inside the 6-helix bundle. So we have concluded from several experiments that in V7A mutant, BVM binds, but in A1V mutant, BVM either binds very weakly or very mobile, or it does not bind at all. And also in both of these variants, the sp1 tail region is inherently much more dynamic compared to the wild type. So this is another unique information we can learn by MAS-NMR, because this information on the binding interaction, they are inaccessible by any other method, as we don't see any global change or overall change in the protein structure. We just see the dynamics are different. So to dive deeper into the binding activity of the small molecule, let's come back to the phosphorus NMR experiments here. So as I mentioned before, my co-worker Roman, he has performed two different kinds of experiments. First, the direct polarization based, where we see both rigid and mobile phosphorus signal from the IP6 molecule, and also in cross polarization, we see signal from only phosphorus where IP6 is rigidly bound. So what's going on in the wild type? Let's see that first. So in wild type, in presence of BVM, we see phosphorus signal from IP6 in presence of BVM. But in absence of BVM in this blue spectra, we don't see any signal because IP6 is not rigidly bound and it's dynamic. We observe the same for the V7A mutant, but for A1V mutant, we see no signal of phosphorus from the IP6, either in presence or absence of BVM. And what does it tell us? It tells that the IP6 is motionally restricted in both V7A and wild type, but IP6 is dynamic in the A1V assembly. So now we know that in both wild type and these variants, BVM governs the dynamic of IP6 in very different way. And in fact, we see different IP6 to protein correlation, phosphorus from the IP6 and proton from the protein correlations, which defines different type of orientation and binding of IP6 in the wild type and in V7A mutant. So IP6 can be probed to detect the dynamic and interaction of small molecule inside the protein assemblies. So with all of this, I would like to summarize what we have so far. So we have a protocol to solve atomic resolution structure of natural abundant small molecule inside the protein assemblies. We have also shown that the power of phosphorus NMR, which can be probed as a time efficient screening tool to understand small molecule binding and activity. And we have also detected novel atomic level interaction of the protein ligand interaction and this tightening of the 6 helix bundle pore, rigidification of IP6, etc. And we have also observed distinct BVM binding and resistant mode IP6 dynamics inside the wild type and BVM resistant variants. And finally, we have solved the atomic resolution structure of protein assemblies in complex with small molecules. So with all of this, I would like to thank all of my members who were a part of this project, all of my group members, our UDNMR facility scientist, and my advisor, Tatiana Polinova, who is here. And this is a long-standing collaboration with University of Pittsburgh, PCHPI Center, Angela Gronenborn, Barbie Pornilos, Owen Pornilos, and Kanil Zadrosny from University of Virginia, Eric Fried from NIH and CI, and Juan Peria and his group at University of Delaware. I would like to also thank all of the funding agencies and thank you all for your attention. So, just one more thing, if you would like to know more about research from our lab, please visit poster 24 by Roman Zadrosny. Yeah.
Right. Thank you, Sucharita, for a beautiful talk. Are there any questions from the audience? Rieko.
Yeah, it's very nice structure and complex formations. If you can tell where the protease should access for maturation and how that is prevented by this inhibitor, that will be nice.
Yes. So let me go back to this slide. So this binding, this is the CA-SP1 maturation switch, which is the junction of the CTD of the capsid and the SP1. So protease accessing from here. And when this small molecule, it binds, it just blocks that pore in a way so that protease cannot access anymore.
Directly block that site.
Yes.
Thank you very much.
So I just have one small question, just to make sure that I understood correctly. So there is a hexamer of proteins, and there is one molecule of the IP6 and one molecule of the inhibitor.
Yes.
So I just was curious if in the process of defining the structural model, so how, when you have the distance restraints, how do you figure out like to which helix a particular distance restraint corresponds to, and I guess what is the conformation
Yes.
of the inhibitor? If you could comment on that.
Yes, thank you very much for this question, because we had taken a lot of time to figure this out, because as you can see, the small molecule interaction, as I am saying, it's an asymmetric binding. So initially I have applied all of the distant restraints to all 6 chains. But then I realized from the violations in the structure calculation that some of the distances which are more than 20 angstrom or 25 angstrom. So I removed them, and iteratively I can understand, okay, these are the chains where it binding like the shortest distance. So yeah, so this is a hexamer of protein, and there is a 2 small molecule, and we have calculated this hexamer of hexamer, and we have taken into consideration the central hexamer, which have all the neighboring interactions.
You don't have to answer this necessarily, but do you have any thoughts about why, I mean, if the molecule is so well localized, why, for example, it's not visible in the crystal or the cryo-EM structures?
So in cryo-EM and X-ray, they apply a symmetry to model those small molecules inside the electron density map. So if you see here over in this figure, so as we can see from here, the electron density, it does not really define the orientation and the binding interaction. So that's why it was not possible. But in NMR, we see direct correlation of these protons from the small molecule to the direct correlation to the protein. And by that, we were able to define the orientation and binding.
All right, if there are no other questions, let's thank Sucharita again for a beautiful talk.
Thank you very much.
So next speaker in the session will be Rongfu Zhang. He'll be talking about selective O17 NMR at ultra high fields, revealing multiple highly stable waters and hydrated phospholipid bilayers.
Hello, hi. Thank you. Oh, so. Working. Oops. Okay. Hi. Thank you. So I'm going to talk about waters, specifically membrane-bound water, and I'm going to talk about the new O17 NMR method. And with this method, it would enable us to identify different types of membrane-bound waters on the membrane, and on the cell membrane, or like lipid bilayers. So here shows you biomolecules solvated in water molecules, and waters are required for the proper folding and function of biomolecules. And we can broadly categorize the waters into 2 types. One with bound water, which related to the waters that close to biomolecules and with reduced dynamics. And the other type with the bulk water that is further away from biomolecules and behaves like more pure water-like, pure water property. And I'm going to refer to these 2 types of waters for consistency, as in the literature, you may find that sometimes people may refer to the bound water as confined water or rigid water or interfacial water, but just to be, so they basically talking about the same type of water that is with reduced dynamics and closer to the biomolecules. And these 2 types of waters are actually in inter-exchange with each other rapidly. And it was proposed to be on the time scale of sub-nanosecond. And we are utilizing the DMPC bilayer as the model membrane system. And as shown here, the lipid bilayer. And the lipid bilayer with the lipid head group and the acyl chain, hydrophilic, hydrophobic, and the water. And the bulk water is away from the lipid surface, with the bound water close to the lipid surface, and they are interchanging rapidly with each other. And if we are zooming on a specific lipid molecule, we will be easily identify 3 type layers that can serve as the bound water binding sites, namely the positively charged choline layer and the negatively charged phosphate layer and the carbonyl layer. And again, they can be interchanged at a sub-nanosecond timescale as proposed. So 0.17 nanomolar is attractive in studying water that associate with biomolecules in general. In that, it's a quadruple nuclei with a spin 5½ and it has a large chemical shift span, which is about 2000 ppm. Makes it sensitive to the local chemical environment and molecular dynamics. However, it does come with some drawbacks. The low natural abundance and small gyromagnetic ratio make it a very insensitive nuclei. And additionally, the second order quadrupolar effect, which are not fully averaged by magic angle spinning, can broaden the solid state NMR spectra and also result in undesired line shape. But we can tackle this by using 0.17 enriched water to enhance the sensitivity. And by going for high field, which for here, we used 35.2 Tesla SCH, corresponding to 1.5 gigahertz proton frequency. Since the second order quadrupolar coupling is dependent, inversely proportional to the magnetic field. So with all that, then what's the difficulty? And there's a fundamental challenge in study membrane-associated water, which is the population difference between bulk water and bound water. Here shows you 2 samples we prepared in the biochemistry lab, one for MS, the other for glass slice oriented sample. For this typical sample, the hydration level is about 50% by weight. And with this 50% by weight, there is a huge difference, population difference between the bulk and the bound water. So if we are to acquire 0.17 nanomolar spectra on these samples, what we get is a bulk water dominant signal. And any bound water signal will be buried and not observable. So how are we gonna observe this bound water signal? If we can suppress the bulk water selectively while maintain the bound water signal, then there's a chance that we are able to characterize the bound water directly with 0.17 nanomolar. So the question becomes how? How are we going to suppress the bulk water signal, but not suppress the bound water signal? To solve this, we need to figure out what's the difference between these 2 types of water, bulk water and bound water. So bulk water, intuitively, is highly mobile, which results in small quadrupolar coupling. And for bound water, it's interacting with the biomolecules, specifically here, the lipid membrane, result in reduced motion and a larger quadrupolar coupling. More specifically, if we compare with the typical eye pulse, it's well known that if the quadrupolar coupling is much smaller than the applied eye pulse frequency, then the eye pulse is non-selective, which means all the transitions is gonna follow in the nutation frequency of nu1. On the contrary, if the quadrupolar coupling is much larger than the applied eye pulse, the nutation frequency will follow this equation here, I plus 1/2 times nu1, which for O17 is 5/2, so it's 3 times nu1. If we to plot the nutation profile against the flip angle, the bound water would oscillate 3 times faster than the bulk water. If we are to choose a small flip angle, such as for the bound water is actually at a 90-degree flip angle for max signal. And for the bulk water, it's reduced to 50%. So we are kind of in the right direction, but it's not enough because it basically you just reduce the signal by half. So how are we gonna reduce it even more? So in light of a recent publication by Ri Chang that featured in the JMR, he introduced the new pulse sequence 10 triple pulse with, as opposed to the single pulse sequence. And how does this work? So basically, the tricks relies on the first 2 pulses. So the third pulses basically serve as a flip pulse for the signal observation. And for the first 2 pulses, here shows you for the first 2 scan of the phase cycling. And if we look at the first scan, for the phase of the first 2 pulses, they have object pulse. And after the first scan, basically the net inference is 0, which means it flip back to the previous demagnetisation. And for the second scan, we use the same phase for the first 2 pulses. For this time, the Z will be scaled by a cosine to theta. And by using opposite receiving phase, we basically subtract the first and the second scan, which result in a net scaled demagnetisation as shown green line here. So if we were to look at the single pulse, the observable NMR signal is proportional to the sine of the flip angle theta. And for this triple pulse, in addition to this sine theta, we have a scaling factor resulted from these 2 pulses, which is 1 minus cosine to theta divided by 2, because it's averaged by 2 pulses, 2 scans. And if we were to plot these 2 nutation profile for single pulse and triple pulse, we notice that for the triple pulse, it decays much faster than the single pulse. And if we were to plot the bound water for the triple pulse as well, and again, choosing the 30-degree angle, flip angle, we maintain the bound water signal at max 90-degree pulse. And for the bulk water, single pulse 50% off. And then for triple pulse, now we reduce the signal to 12.5%. It's pretty good, but it's not enough. So what do we do? Oh, sorry. So since the trick lies on the first 2 pulses, so what we do, we add additional 2 pulses, and then we add additional 2 pulses. Basically, we add in 2n plus 1 pulses. And then now, the signal is scaled by 1 minus cosine 2 theta to the power of n. N can be 0, 1, 2, 3, etc. And if we were to plot the nutation profile again, now what we get is we get the even 0, 1, 2, 3, and again choosing 30-degree flip angle, and by a simple calculation, now we can down to 7 pulse is less than 1% of the original signal, and this is good enough for us. So with that in hand, Now we need to go into do some real work, which is the magnets here, which is the 35.2 Tesla, and the home built MS probe and oriented NMR probes to obtain the signals. And so we first measure the signal and to verify the suppression, how well it compared to the theory. And as shown here, when we set the N to 0, 90, 22, instead of using 30 degree, we use 22.5, which add additional suppression, but also decrease the bound water signal slightly. And this is what the suppression value we get. And if we compare to the theoretical suppression, it agrees very well. Which means this method works experimentally. So now we can go to the lipid membrane system, hydrated lipid bilayer. And we first measure the T1 relaxation time of the bulk water. And we got a value of 3.7 milliseconds. And you usually refer to bulk water as pure water like. So we measure the pure water relaxation time as well, without lipids. And the bulk water is actually smaller than the pure water, which suggests that this bulk water is in fast exchange with the water molecules in the lipid head group, outer layer, where the motional freedom is reduced. Means this bulk water is not free as pure water collectively. And we attribute to the top layer of this phospholipid head group. And we noticed another portion of water that has a chemical shift of 0.17 ppm when we do zero crossing on the bulk water signal by setting the tau D to 2.6 milliseconds. And we measure this T1 relaxation of this small piece of signal. We get a different relaxation time, 2.5, compared to the previous 3.7. And this smaller chemical shift difference and slightly shorter T1 relaxation time suggests the existence of a small portion of water that is not in fast exchange with bulk water. Because if they are, then we will not be able to identify this species of water. But they are still relatively mobile because the relaxation time is still fairly slow and the chemical shift is small. We attribute this to the water that associated with the phosphate layer and termed as confined water. And we move on with doing some aligned sample as well. So it's aligned lipid bilayers. And what we see is for the bulk water, we observe a small residual quadrupole coupling, about 6 kilohertz, which is relatively small compared to the large quadrupole coupling of the O17. And this agrees with the bulk water, means it's not pure water like. There's a small RQC existence in the bulk water. Interestingly, we also observe a small additional RQC, which is about 2.7 kilohertz. And this has a smaller population. Again, agrees with the MS results with the confined water. So far we have been identifying the waters that has fairly dynamic and small quadrupole coupling constant. So what about the large quadrupole coupling constant water, if there is any? Oh, sorry, there's another. So I've shown about this. So basically now we set the theta to 22.5 degree, which means if by this degree, we suppress the bulk of water as previously suggested. And we use n equal to 1 on the 1.5 gigahertz magnet with the n equal to 1, with not very okay enough separation of the bulk water, but maintain the bound water signal. So here shows you the blue, we see a bump. And then when we set the flip angle to 60 degrees, if this is resulted from a large quadruple coupling water, then the 60 degree equals to the 180. We should see a null signal as indicated by the purple here, suggesting it's resulted from a large quadruple coupled water. And we move to n equal to 3, which is 7 pulse. So now we sufficiently suppress all the bulk water signal, and we resolve a very nice O17 resonance with a chemical shift fairly large away from the bulk water, which is at 0 ppm, and it's minus 11.6 ppm. If we compare to the previous 0.17 ppm, this is pretty large chemical shift difference. And to observe the second order quadrupling effect, we go down to the low field, about 800 megahertz. And we observe the same peak, but it's significantly broader than the ones on the SCH, demonstrating the powerfulness of a high field for quadruple nuclei, such as O17 study. And here also shows the second order effects, the line shape resulted from the second order quadruple coupling. And we can also measure the T1 relaxation of this population of water. And it's an order smaller than previous ones, 3.7 millisecond. So here suggests the bound water exhibits large quadrupolar coupling, and we attribute to this coupling region, which has a very confined space hindrance and allow a lot rigid water existence. To conclude, with the method O17 bulk water suppression, we were able to identify multiple different type of states of water population. And these waters can be stable on a millisecond timescale, which is quite different from previously proposed. And this new method can also be attributed to other type of biomicromolecules, such as proteins and DNA. Yep. Sorry. I need to thank, there are lots of people without this many people that this work can't be done. And I want to thank the team for the opportunity to conduct this type of study, and Rui Jiang as a very good collaborator, and Peter and Wenping when we have a problem with the probe. That's the people we come to, and Ilya for when we have a field regulation problem, and that's the person we come to, and also the grant support from NIH and ASF. Thank you.
Right, thank you, Rongfu, for a very nice talk. Looks like Jeff has a question, and then another one right after.
Lovely talk. Really enjoyed that.
Thank you.
About nearly 50 years ago, Tak Kuntz did some experiments on protein solutions.
Okay.
When he froze them, he saw there's a residual sharp water line with an area that's proportional to the protein concentration. And he interpreted that as indicative of a mobile interfacial layer, even in the frozen solution.
Okay.
So not totally bound to the protein and not bulk.
Yes.
My question is, can you freeze your bilayer samples and look for similar effects in the O17 spectrum?
Yeah, that's actually a good question. And we did not try that. And we certainly can. But for this, most of the studies here, we try to maintain it above the physiological, like the phase transition temperature, such as that we were able to align, like the DMPC, we have to maintain about 22 degrees to align the lipid bilayer. And we also try to study this physiological condition. But this does have literature suggesting if you are going to very low temperature or below the phase transition temperature, for sure, the population of the water around the lipid head group will change. And I'm certainly think the dynamics will change as well. Like the population that's suggesting will be reduce the water molecules numbers around the lipid head group region when the lipid is in different phases. Yeah, so, but that's an interesting approach. We may try, yeah, permitted if Peter can, because the VT has to be able to cool down to frozen, and we are pulsing very fast on the 017 because the signal sensitivity is low. So the delay is very short, and there may result some sort of RF heating, and yeah, and so we need to have very good cooling.
Hey, nice talk. So I had a question on, have you tried measuring the exchange rate, like directly using any technique?
Exchange rate?
Between the bound and the free water. Have you tried measuring the exchange directly?
No. And actually, we know the exchange rate, it must be on the NMR time scale.
Right.
We don't measure directly. It may be slower than that, but at least need to be on a millisecond time scale. Yeah, because if not, then we won't be able to identify those species, right?
Sure.
Yeah.
The follow-up question was, have you tried doing any like direct proton oxygen type experiments to selectively observe the bound water at all, or does that not work?
Yeah, actually, that's a good question. We did try, and yeah, so far we don't have good results on that yet. And
so it's probably the case then that it's still dynamic, and that's
yeah,
probably
the transfer is really difficult
right.
between these two, and I think yeah, that's the goal actually. Yeah, thank you.
Right, I think in the interest of time, I think we should move on
Okay.
to the
Yep,
next
okay,
speaker.
yep.
Let's
Thank
thank
you.
Rongfu again for a
Right.
very nice talk.
Thanks.
The next presentation will be by Tom Popp from Rutgers University, and he'll talk about 3D printed centrifugal sample rotor preparation devices for more than 100 kilohertz MAS NMR.
Can everybody hear me now? Okay, thanks. So the ability to spin samples at 100 kilohertz MAS has enabled the collection of very high resolution solid state NMR spectra of biomolecular solids. However, the preparation of these rotors for these experiments can be quite challenging. So today I'm going to tell you a little bit about the work that we've done in developing 3D printable centrifugal packing tools for these rotors for these experiments. So our main problem when doing this is that biomolecular solids have properties that make them squishy. We've got two very commonly encountered forms of biomolecular solids in the study of proteins. Liposomes are a common vehicle for membrane proteins. And when we study proteins alone, they're often encountered as nanocrystals. Liposomes, when centrifuged, have the texture of like a gel-like pellet. And protein nanocrystals are more like a slurry or a suspension than a nice dry powder. And so these are a lot more challenging to put into MAS rotors than dry powders. And so as a result of that, there has been a precedent in the biomolecular solids community to develop centrifugal packing tools for MAS rotors of any size. These take on one of two different conformations. One is a sample transfer tool, where we take a pellet that's been pre-centrifuged, and then we just basically transfer it at low speed, low centrifugal forces into the rotor. And that's like this type of device here. And in fact, in the NuCoop lab, we use this one for our 1.6 millimeter rotors. But then we also have another type where we directly ultracentrifuge solids out of solution in the tool. And these are these ultracentrifugal devices that you can see here. These are typically machined out of durable plastics like PEEK or Delrin, which can withstand high centrifugal forces. And our challenge when designing a tool to do this for 0.7 millimeter Bruker rotors is that these rotors are just really, really small. They have a mass of around 6 milligrams, and their sample volume is only on the order of 1/2 of a microliter. So they're so small that even packing dry powders into them can be really challenging. Bruker has developed a protocol with a suite of packing and handling tools that makes this process really elegant for dry powders. And you can see in these photos here, some of the tools and some of the actions that you'll be taking while doing this protocol. This protocol is such that you're never ever going to be touching the rotor with your hands, which I think is optimal because these things are small, they fall on the floor and they can break, so you don't want to lose them. But just for a sense of scale on this tool, you can see that this red arrow is pointing out the 0.7 millimeter rotor within the tool. They are almost really hard to see, like even just by eye. And so this protocol wasn't really designed for biomolecular solids. And so for a couple years in the NuCoop lab, we've been packing our 0.7 millimeter rotors with sort of a manual protocol where we centrifuge into a pipette tip, and then we transfer manually that packed material into the rotor by inserting into the pipette tip. This had some drawbacks though. You can see here 16 different NH2D spectra of microcrystalline protonated GB1, each taken at 105 kilohertz MAS spinning rate and on our 800 megahertz instrument. I would say that this process worked maybe about 25% of the time. These spectra up at the top have the resolution and properties that we expect for well-resolved, good, well-packed, well-hydrated microcrystalline GB1 at this spinning rate. But the other times, they did not turn out nearly as well. And I want to point out that there's sort of a spectrum of bad spectra that we get when packing sometimes. There's very obviously something that went wrong in these ones. Maybe it's dehydration and probably dehydration. But in this one, sometimes the sample didn't even transfer properly into the rotor. And so this one was mostly water. But I think what's a bit of a scary thought is that these ones look almost okay. If we didn't already know what microcrystalline GB1 was supposed to look like at 105 kilohertz MAS, and we were studying a protein that had not been well studied, we might say this is good enough resolution and let's proceed forward. And we might not realize that we're looking at something that's suboptimal. So when we design a packing process or a packing tool, we want to make sure that we're so confident in this tool and in this protocol that we can get the best spectra every single time. So yeah, those are the four that are the best. So we set out to make a centrifugal packing tool that can efficiently and reproducibly transfer biomolecular solids into 0.7 millimeter Bruker rotors. Our main design criteria for this effort were consistency in our protocol. We wanted to be able to interface nicely with this protocol and tools and packing tools that Bruker has already developed. We want to have it to be reproducible. We want to get the best spectra every single time and be confident in every rotor that we pack. And then we also want our tool to be adaptable. We want it to work for different centrifuges and maybe even slightly different sizes of rotors or different changes or anything that we want to have. We want it to also be just customizable, basically. And to do that sort of adaptability, we use 3D printing, and we 3D printed with a Formlabs Form 3 SLA 3D printer, which is a commercially accessible benchtop 3D printer. And it has a spatial resolution of 25 microns in X and Y, and in the resins that we're using, typically has more of a Z resolution, about 50 microns, but still really, really good. We can print very small features with these tools. The resin that we're using is a gray thermoset resin that has properties similar to PEEK or Delrin when UV cured. When thinking about designing our centrifugal packing tool, we want to think about what is the platonic ideal of a centrifugal packing tool. And so here's our platonic ideal. It's sort of featureless, but that's the point. We're just trying to think about what we want. And what we want it to do is fill our rotor completely with appropriately hydrated sample so that when we pack it, put it in the magnet, we're happy. That means addressing areas at the entrance and exit of the rotor, because what we don't want is to leak our very valuable sample that we spent a lot of time preparing, or to improperly fill the rotor regardless, even if there isn't a leak, because that can lead to underfilled rotors, which is just low signal, and we want to have as much signal as possible. So let's say we address our leaks. We also want to make sure that our capping process doesn't result in partial or complete dehydration of the sample, because we can run into problems, as you saw before, with our NH2Ds. So here's what we designed for our low-speed centrifuge for packing samples into 0.7 millimeter rotors. The tool is comprised of several 3D printed parts that connect and align all of the components in this assembly. And then this whole assembly can be placed into a 50 milliliter centrifuge tube and then spun down to transfer the sample. This tool is modular, so we can use it for ultracentrifuge tubes. And this version of the tool we would use for our sedimented liposomes that we've pre-sedimented in an ultracentrifuge. But we can also use it for protein nanocrystals, and this is the type of tool with this pipette tip already embedded that we would use for our protein nanocrystals. We prevent leaks in the base of the tool with an O-ring, and then because of the low speed of this centrifuge, we can insert the rotor into the tool with the cap pre-installed, which also helps prevent leaks. And in this nanocrystal tool, we also have this pipette tip, which serves as an additional leak prevention mechanism at the top, where the rotor is partially inserted into the pipette tip. And we're able to do that because we have a pipette guillotine that we've developed that cuts the rotor at exactly where you want it. You just take a razor, put it up against this, this bar here, and then cuts off the pipette tip to the point where the opening is now 0.7 millimeters. And you can then, when you assemble the tool together, insert the rotor into the pipette tip with a snug fit. And of course, our designs have evolved over time. We've gone through different iterations of the tool and used different 3D printing resins. And so these images show generally how our packing process works, even for our newest version, but it's just with an older version of this tool. We have, and you can see how it neatly interfaces with our Bruker tools. We have the blue grabber tool for the 0.7s, which then also aligns with the base, and we can then insert the rotor there. When we put the whole thing together with our pre-guillotined pipette tip, you can see that we then insert the rotor into the pipette tip quite easily. And the crucial element of this is that everything stays aligned, so we're not worried about the pipette tip not hitting the rotor and then cracking it or something like that. Everything stays concentric. We can put everything together with our sample in the Eppendorf tube here, spin it down, and then we can look at the result. And in our pipette tip here with the rotor installed in the bottom, we have a large water column and a packed pellet of nanocrystals that is overflowing out of the rotor, meaning that the rotor's full of nanocrystals, and this water column means that those nanocrystals are well hydrated. Now, this interface between the pipette tip and the rotor is not 100% perfect. It's going to leak a tiny bit. And so that's where the O-ring does its job. There's a little bit of water there, but the O-ring provided a backstop against that water column. I want to point out that there is precedent for this sort of transfer process using laboratory consumables like centrifuge tubes and pipette tips, but our process is a lot more consistent, and it also works for 0.7s. Previous versions, you're chopping different laboratory consumables at various places and parafilming them together, which can be inconsistent. And so our process enables us to transfer material into our rotors without ever needing to grab the rotor at any point with our fingers or anything like that. We've also developed a version of this tool for the ultracentrifuge, which to our knowledge might be the first ultracentrifuge packing tool that is 3D printed. But the basic idea in the assembly process is the exact same as our low-speed centrifuge tool, where we have a bunch of components that assemble together, and we have the pipette tip involved. The sample goes into this funnel here, and then we can sediment our either our slurry or solution that we want to sediment some solids out of solution into our rotor. The main difference with this tool is that it's a smaller form factor for the ultracentrifuge rotor, but that we also don't put in the bottom cap because we don't want to crush the cap under the high G forces. But because of that, we put a little disc of parafilm in at the bottom to prevent leaks through the rotor in addition to the O-ring. We've tested this tool up to 200,000 Gs with no visible deformation of the plastic, but there are even thermoset resins that we can use the 3D printer with that can withstand even higher mechanical stresses. So those could potentially push this to even higher G forces if we're interested in doing that. So when we're using either of these styles of tool, we're going to need to take the rotor out of the tool, and then we need to cap it. What we do to prevent dehydration at this stage is we do the entire process of capping in a humidified glove bag. Getting a humidified glove bag is easy. You get a glove bag from Sigma Aldrich, a small humidifier, and a humidity sensor. You turn on the humidifier for 10 to 20 minutes, and then you have a bag with 95% relative humidity. This is important for us because our lab routinely gets down to around 10% relative humidity sometimes in the winter. And so that is a serious contributor to whether or not our samples are going to be dehydrated, especially when we have very small samples that can dehydrate quickly. And so this protocol makes sure that this sample that you've worked for so hard up to this point can make it into the magnet unscathed. So now let's take a look at the results of using these packing tools with some NMR. So we're going to use the same mechanism to check them out again, fully protonated GB1 at 105 kilohertz MAS. And comparing to our manual packing protocol, which I guess had about a 25% success rate. And in the red, we have a representative example from those 16 NH2Ds before. We've now used our ultracentrifuge tool with the glove bag packing protocol about 4 times now, and we've had 100% success across all those times. We get a spectrum that looks like the blue one every single time we do this protocol. So this gives us a lot of confidence that whatever we put in the magnet is exactly what we want to be seeing. I want to point out that no matter how hard you centrifuge something in, even if you're using an ultracentrifuge, which is at 200,000 Gs, spinning a rotor up to 105 kilohertz MAS produces a G-force of 11 million Gs at the inner wall of the rotor. So that sample that was packed in there pretty tightly to begin with is now going to be packed even more tightly up against the walls, leaving behind a central water column or a void, which has been noted before. But what you can do is you can, with our protocol, uncap the rotor in the glove bag, pack more crystals back in, and then put it back in the magnet. And this results in a 20% increase in ambient proton signal that we have seen so far. This isn't DNP, it's not a huge enhancement, but we really like to get extra signal for free. So especially with an easy enough protocol to do so, I think it's worth it. We've also begun to explore using these tools for packing liposomes into 0.7 millimeter rotors, and we're excited about the results because it looks like we're getting efficient filling of the rotors. And we're encouraged by this because we're in the process of studying lipid-lipid interactions, especially in mixed lipid liposomes, as well as membrane protein to lipid interactions in these membranes at fast MAS. Of course, the most exciting part for us is that these tools are accessible to and available for and beneficial for any lab studying biomolecular solids with solid state NMR. And we want to embody this vision put forth in a recent review by Rachel Martin's group and echoed by many, many others in the NMR community of sharing and developing tools for magnetic resonance experiments in an open and collaborative way. So that means if you have a 3D printer and you want to print these, we're happy to share part files with you. If you don't have a 3D printer but you want to use these, we can send them to you. We'll print them ourselves and send them to you. And if you have a different centrifuge or different rotor size, maybe a 0.75 or maybe something a little bigger like a 1.3, we can very easily modify these tools and adapt them to a specific, whatever specific setup that you have. And with that, I'd like to acknowledge and thank Andy, Jackie, and Ashley for their work with me on this project. Andy has a poster, number 20, so be sure to come check that out. Ashley also has a poster, number 17. So stop by and learn more about what we're doing in the NuCoop lab. I also especially like to thank my 2 undergraduate students, Rashawn Green and Branson Matchett, who have been working with me on the design and optimization of the ultracentrifuge tool. I want to thank the NIH for funding our lab's research, and I also want to thank the Arnold and Mabel Beckman Foundation for funding me to be able to make chemical instruments for NMR spectroscopy. I'm looking forward to having more conversations about rotor packing and sample preparation, all things like that. And please do reach out if you're interested in using these tools. Thank you very much.
Thank you very much, Tom, for a very nice talk. I'm sure there'll be lots of interest from the community in using the tools. The question from Rongfu.
Hi. You mentioned you start to work on the liposome packing, right? And have you tried any larger, in terms of water contents, and then have you observed any leaking,
leaking of the tool?
Yeah,
or,
because
Up to this point, we have not used the ultra-centrifuge packing tool for liposomes. So that's something I didn't point out. What we've been doing with them is to use our low-speed centrifuge tool for packing liposomes. And as you can see, there's less leak prevention in this version. We don't have the pipette tip and everything for this one. Our liposome pellets are typically viscous enough that, and we usually have them large enough that we can kind of overfill the entire area down here and having a bit of leaks. So yeah, it's a bit messy. It's not as nice, and we do want to optimize that a little bit more, but we do end up with efficient and full packing of our rotors.
Yeah, very interesting.
I just had one question to clarify. So in the one that you don't use the ultracentrifuge for, do you still cap the rotor in this glove bag with the controlled humidity?
Absolutely. Yeah, I mean, I think that may be even one of the most important parts of this protocol is doing everything in that glove bag. We can absolutely ensure that we're not dehydrating our samples.
Okay, with that, let's thank Tom for a very nice talk.
Thank you.
So the final speaker of this morning's session will be Galia de Belucina from UCSD, and Galia will talk about reevaluating polarization agent design for in-cell DNP applications.
Hi, everyone. Can you hear me? Yeah, okay, great. My apologies for the technical issues. I guess going back to in-person talks, we also going back to technical issues. I'm very grateful to be here today. Thank you to the organizers for the opportunity to present today and also to be here in person and to see all of you after 2 years. So today I would like to talk about our work on targeted DNP with the idea to use it in cells, and also our thoughts and experience when it comes to different polarization agents for this particular application. So I don't have to preach to the choir here. We all know that NMR spectroscopy can be a very powerful tool for structural biology in cells. However, in order to really unleash the power of this technique for structural biology in cells, we have to solve what I like to call the 2 S problems of NMR in this context. So the first S problem is sensitivity. We have to look at very tiny amounts of proteins in cells. So we really need a lot of sensitivity in order to be able to see nanograms and picograms of material in the cell. And so I tend to think of this as a physical chemistry problem where spin physics and techniques like dynamic nuclear polarization can really help us achieve this sensitivity. On the other hand, the second S problem for in-cell NMR is selectivity. How are we going to even select our favorite molecule from the cellular environment among all the other molecules that are present there? And I tend to think of this as a chemical biology problem because chemical biologists for a long time have worried about and developed tools to select specific molecules in the cellular environment. And so my group combines the tools of physical chemistry together with the tools of chemical biology to develop targeted DNP approaches for in-cell applications. So when it comes to DNP NMR spectroscopy in cells, there are currently two approaches that people have used in the literature. The first approach is an approach that's also borrowed from solution NMR, solution in-cell NMR spectroscopy, where you can prepare your favorite protein in E. coli, where you can C13 and 15 label it, and then you can use a technique called electroporation to put this protein inside the cell. And we typically, for this, you have to use a lot of polarization agent because the polarization agents that we have at the moment are actually not cell permeable. And as Kendra Frederick has recently shown, one can actually use electroporation to also increase the amount of polarization agent inside the cell. And so this has its advantages. It allows us to achieve selectivity through the isotopic labeling. And then we have sensitivity through the dispersed polarization agent inside the cellular environment. However, this always means that we have to introduce our protein from the outside. And often we end up with a huge amount of protein in the cell that's not biologically relevant. And also the act of electroporation can damage the cells. And when the biradical is dispersed everywhere in the cell, we can also get a lot of background. So for this reason, a lot of people, including us, have really thought about this idea of targeted DNP. And the idea of targeted DNP in the cellular environment would be a situation where you have selected your favorite protein and you have found a way to attach one polarization agent to your favorite protein. And this polarization agent will be responsible for increasing the signal of your favorite protein and not really increasing the background. And that will give you sensitivity and selectivity in the same step. And one big advantage of this potential approach is that you would be able to look at proteins that are endogenously made in the cell and potentially looking at more endogenous level concentrations and more biologically relevant concentrations in cells. So just to kind of highlight a few things that we have observed about targeted DNP. One is that targeted DNP may offer advantages at faster MAS. And also, we can easily control how far the polarization travels in these types of targeted systems just by looking at the buildup times for the enhancement. So, so there's certainly some advantages. And also one big advantage that is not on this slide is that this method certainly outperforms conventional DNP approaches at low concentrations. So the way we do targeted DNP is a little bit different from everybody else. So what we do is we take advantage of the molecular biology techniques that allow us to do genetic manipulation of biological systems inside the cells. And this allows us to install a natural amino acids inside the cells. And so in particular here, we have an example of the protein ubiquitin where we've attached the polarization aid, we've attached the natural amino acid norbornene lysine at a specific well-defined position in the protein. And then we have taken totopol, not because it's the best polarization agent we have available, but because it's the easiest to synthesize. And we attached a new chemical group to it called the tetrazine. So this tetrazine moiety finds the norbornene moiety inside the cellular environment, and the two react very selectively in a reaction that's called a bioorthogonal reaction because they don't touch anything else in the cell. And what are some advantages of this approach is that you don't need cysteines anymore to do your targeting. Some of you are used to doing that, for example, by targeting PRE agents or or even fluorophores to your favorite proteins through cysteine chemistry. Well, here you can keep your cysteines to do whatever they need to do. You don't need to worry about them. And this approach, because it's genetically encoded, it allows us to do a natural amino acid incorporation in both bacterial and mammalian cells. It is in principle applicable to any protein of interest. There's no synthesis required other than your polarization agent, but it's the same polarization agent for everything else. And as I mentioned earlier, it can allow us to do DNP of endogenous proteins. And since some of you are not familiar with this technique of a natural amino acid incorporation, it's also called amber suppression. It's actually a technique developed by Peter Schultz's lab at the Scripps Research Institute, just down the road from us in La Jolla. And it is very easy to implement. So what you need is 2 plasmids and the unnatural amino acid. So the first plasmid is the plasmid of interest, where you have a mutation at the particular position where you want to install the unnatural amino acid. And then you need the second plasmid, which is for an orthogonal amino acid tRNA synthetase, tRNA pair, that can recognize your unnatural amino acid and help load it on the ribosome. And then we need the unnatural amino acid. And then once you have all these components, the cells do the rest for you. So the cells produce an mRNA that carries the mutation. So typically we use the stop codon, the amber stop codon UAG to encode for the unnatural amino acid. And typically the ribosome will just stop there and not translate. However, because now we have a new tRNA that carries the unnatural amino acid and that can recognize this stop codon, the ribosome can now install the unnatural amino acid at that particular position. And then we end up with a polypeptide chain that contains your favorite, that contains the unnatural amino acid at your favorite position. And so whenever I've submitted this for grant proposals and also I've talked to some of you about it, I get the reaction is always the same. This is a nice idea, but it never works for NMR. We have by now shown in at least 6 different systems, both in mammalian and bacterial cells, that a natural amino acid incorporation can work well, even under isotopically labeling conditions. We can, so far, any protein that we've wanted to make with an unnatural amino acid, we have been able to do so. However, not every position we've chosen has worked. So if you are ever trying this technique, a good idea is to actually try a few different locations for the unnatural amino acid. And so far, we've always been able to find a location that works well. And so we have been able to apply this to several different proteins, and these are targeted DNP spectra of 3 different proteins at concentrations of around 50 micromolar or about 5 to 10 microgram of protein. And so here the enhancements with targeted DNP are around 5 to 10. However, still enough for us to see some signal. And we're confident that we can do better when we have better polarization agents. Remember here, we're still using Totocol. We have also applied this technique to do targeted DNP in bacterial lysates. So here, for example, we've targeted ubiquitin in bacterial lysates that were dialyzed to remove reducing agents. And just to kind of cut a long story short, what we observed here is a total enhancement of 10. The background was enhanced by a factor of 2, but the selective enhancement on ubiquitin was a factor of 5. So we have a factor of 5 selective enhancement of a background. And again, we are confident that we can do better now that we understand much more about the polarization transfer mechanisms of these in these types of conditions. We are also currently applying this technique to study membrane proteins in bacterial cells. This is a collaboration with Francesca Morassi's group. They're very interested in this protein called AIL, which is expressed in E. coli. And here we're looking at intact E. coli. We have actually placed the polarization agent right here on the extracellular membrane. And we are currently doing DNP with this system, but here I want to show you the EPR, which shows that we can actually successfully attach this polarization agent to the protein in intact cells while the protein has the unnatural amino acid on the outside. And also natural amino acid incorporation works in mammalian cells. Here, I am showing fluorescent proteins. I apologize for this, but it is actually very easy to see what's going on. So we have this system where we have a red fluorescent protein, which tells us that the mammalian cells got the plasmid that we transfected them with. And then the green fluorescence tells us that we actually installed the unnatural amino acid successfully in this context. So installing the unnatural amino acid works really well. We have the technology, we have the protocols to do this. Attaching the polarization agent also works reasonably well, provided that we can get it into cells. However, our issues for continuing this work much more aggressively into mammalian cells or in cells has been the polarization agents. So, this has recently become quite a hot topic in the literature. So several groups have looked at the stability of polarization agents in cellular environments. So we know that there is reduction in the cellular environment. And so the ideal polarization agent that we need is something that gives us large enhancements, something that is stable in cells, and also something that is cell permeable. And for targeted DNP, we also have to worry about a couple of additional constraints. One is that it actually has to be efficient at low concentrations. And the other one is that it needs to be able to undergo fast bioconjugation. Okay, so we set out to explore and a little bit focus on the stability in cells. And we were inspired by work done by synthetic chemists and EPR spectroscopists looking at the stability of nitroxide radicals in reducing environments. And so in this paper, they showed that if we take this 6-membered piperidine nitroxide, tempo-based, it actually gets reduced very fast in ascorbic acid. So this is this gray trace over here. Now, if we go from the 6-membered piperidine ring to a 5-membered pyrrolidine ring, then the stability goes up here. So this is this black curve over here. And now if you go from dimethyl substitution to diethyl substitution, like over here, this is the red trace over here where it appears that in ascorbic acid, you really maintain your stability for several hours. So what we thought was, okay, let's try to make some polarization agents that are based on these 5-membered ring nitroxides. And we very hard tried to make this one, turn it into a totopole-like molecule. And we even had a name for it. We called it spider-pole. But it turns out we are not a synthetic chemist. So at least for us, this was a very difficult molecule to make. But we were able to make the dimethyl molecule. So this is very much like totopole, except that now we have 5-membered rings instead of the 6-membered rings. And so we call this molecule popopole. Okay, so now let's take this molecule and compare it to several polarization agents that we know. So here we are comparing popopole, totopole, amupole, and asympole POC. And so first we looked at reduction in ascorbic acid by EPR. And so here you can see the reduction in substoichiometric amounts of ascorbic acid and stoichiometric amounts of ascorbic acid. And you can see that popopole outperforms the other polarization agents under these conditions. Asympole POC, because it has one 5-membered ring, also does better than amupole and totopole. Okay, and so here are also the EPR spectra over time, and just wanted to focus on the asympole POC one, where you can see that at the end, it looks very similar to the popopole one, meaning that the 6-membered ring nitroxide got reduced while the 5-membered ring nitroxide is much more stable under these conditions. So then we looked at DNP enhancements. So these are enhancements in DNP juice, no cells, no lysates here, just of proline in DNP juice, first with high concentrations of polarization agent like we are used to, 15 millimolar. So here a good one of the take-home messages is that moving from 6-membered rings to 5-membered rings, the enhancement stays the same. So popopole and totopole have very similar enhancements. And under these conditions, amupole seems to be the best polarization agent, but again, with the caveat that it is very prone to depolarization effects. Now, things become more interesting when we move to low concentrations. So here we are looking, these are the enhancements at 15 millimolar, and these are now the enhancements at 1 millimolar polarization agent, again with proline. And so here you can see that amupole and asympole POC become very similar in terms of enhancement. And actually, if you look at the total signal that you're getting, asympole POC seems to perform much better at these low concentrations compared to amupole. And popopole and totopole still remain very similar. Okay, so then we wanted to look at what is the situation in lysates. So these are mammalian cell lysates. We're following the reduction of radical by EPR. And here we have 1 millimolar of radical in these studies. And so here under these conditions, now you can see that popopole and asympole POC are actually very similar to each other. They have very similar reduction rate, while amupole and totopole are much more sensitive to the reduction in these cell lysates. And then we also did similar experiments, but now we are looking at the DNP enhancement as a function of time, as incubation at room temperature. And so here we had to use a little bit higher concentration of polarization agent because we actually had to measure our enhancement. And with 1 millimolar, it was very hard to get a good number. So in the presence of 2.5 millimolar polarization agent, these are the enhancements as a function of time. So this is true enhancement. And you can see here that asympole POC always gives us consistently higher enhancements compared to the other molecules. And so it's not that asympole POC, it's actually its rate of reduction, it's as fast as popopole or even faster. However, because it starts with this much higher enhancement, even when it gets reduced by 50%, we still have enough in there to get good enhancement. At the same time, pov-pol, amupol, and totopol here really are very similar. So this tells us that really this 5-membered ring on amupol is very important, and it can be very helpful in reducing reducing the dependence on reducing conditions. And it is currently probably the best polarization agent to use for in-cell DMP. And finally, I want to emphasize that, you know, even though these are good molecules that can give us good enhancement, none of these molecules are really good for cells because they're not cell permeable. So here we did an experiment where we incubated cells with polarization agent, then we spun them down, and we actually looked at the presence of the polarization agent in the supernatant. So if the polarization agent got introduced into cells, then the concentration in the supernatant would be reduced. And we actually did this experiment by HPLC rather than EPR because we want to deconvolute uptake of the polarization agent versus reduction by cells. And so what you can see here is that pov-pol, amupol, and asimpol POC are all, they don't really get into cells. Totopol seems to be reduced in the supernatant, but at this point we don't know if this is because it actually gets stuck on the cell membrane or whether it actually gets into the cells. So this is a little bit harder to tell with this type of experiment. Okay, so what is the take-home message? So the take-home message is that currently asimpol POC outperforms all other tested polarization agents when it comes to cellular environments. And it works well at low concentrations, and despite actually, it still gets reduced. But because it starts with this much higher polarization, much higher enhancement, it still gives you enough even after an hour to have an enhancement of 20 or 30. However, moving from 6-membered polarization, 6-membered nitroxide, 6-membered ring nitroxides to 5-membered ring nitroxides has beneficial effects. And we should, as a community, try to think of incorporating them more into our future polarization agents when it comes to applications in cells. And cell permeability is still a problem. So with this, I would like to end and thank my group for really great experience. It's been 5 years. My first graduate students are actually about to graduate. And the person who did a lot of the work here is Bryce Ackerman, also helped by Nasreen and Sirish. And BJ was our synthetic chemist who recently left the lab. Thank the funders and also thank you very much for your attention. And before we go to questions, I just want to highlight this tutorial that we wrote for JMR Open. It was actually written by my undergraduates, Evan and Eleanor, who are also graduating. And it's a chemical biology primer for NMR spectroscopists. So if you want to learn about inteins and a natural amino acid incorporation and how to do it for NMR spectroscopy, please take a look.
Yeah, we have time maybe for one quick question, because I think this ultra high field workshop is supposed to be coming and we had some technical issues, etc.
Sorry.
Very interesting talk, Galia. So yeah. I had a question on, so you said that the cell permeability is low for most of these radicals, but at the same time, what's then causing the reduction? Like, is it their interaction with the surface or sorry, the cell membranes?
So these experiments were done in lysates. So there, the polarization agent is exposed to the cellular environment, to the reducing agents. We are currently doing experiments with intact cells. And the struggle there, just like Kendra has shown, is having good protocols to not kill your cells and let all of these components come out and reduce your polarization agents. prematurely. But sometimes, yeah, just mostly if you keep things outside on the membrane, there's not much reduction.
Okay, thank you. Also, what do you think is the reason for the fact that the 5-membered rings are a little less sensitive to this effect? Because it's a bit counterintuitive when I think about the angles in which like 5-membered and 6-membered rings are present and potential interaction with reducing agents.
I think they're just better sterically protected, especially when you start adding these diethyl groups. There's just better sterics under these conditions. Yeah, at least that's the explanation I have right now.
Thank you.
Okay, last question from Hari.
And I'm happy to answer questions outside too. Yeah.
Lovely talk, Galia. Just want to pick your brain on what are the advantages of your method of introducing an amber codon vis-a-vis synthesizing a peptide with an unnatural amino acid and intine ligating it to any protein that you want.
Oh, that's so much more work.
Really?
Doing synthesis, yeah, and
Because
then
you can create the same
inteins,
peptide,
yeah.
right? But that's just an
Yeah,
addendum.
sure,
It could be a
but-
5-amino acid peptide that harbors the unnatural amino acid. And
Yeah.
all you need to do is that intine it to your ligand. And there you don't need to worry about the yield where the amber codon gets
Yeah, but then
interpreted as a stop codon.
you still have to worry about different construct with the intine every time. So, I mean, it may work, you know, also more generally. However, this is the least perturbative way to do it. And the yields are not as low as you may think. It's just a matter of trying it and playing a little bit with the conditions and your plasmids. And if any of you are thinking about trying this, we have a lot of tips and advice. So please let us know if we can help.
All right, with that, sorry, I think it will have to be after the session with your question, but I would like to thank Galia and all the speakers in the session for very fascinating talks.